My name is Minerva Contreras, and I'm a PhD candidate in the lab of Dr. Nicola Allen at the Salk Institute. Consciousness is everything we experience, and everything we experience must be first processed by our brain. Is there a physical property to consciousness? If we didn't experience it with our brain, did it really happen? Or conversely, can our brain experience things that didn't really happen? What is reality? And is it even real? How does our brain help us understand the outside world or our experiences? You may be familiar with neurons, the most famous type of cell in the brain. Neurons interact with one another, forming the circuitry that help us transform an image into information that our brain can understand. In simpler terms, our brain, with its neurons, forms pathways which are like roads that transmit information. Information transmission happens across neurons in what is known as synapses. Turns out, there is much more happening at synapses than we previously knew. Scientists discovered that a different cell type, the astrocyte, contacts synapses. Astrocytes are star-shaped cells that contact synapses, help them form, influence what happens there, and can even regulate how much a synapse can change throughout our lives. Wait, why would a synapse need to change? Well, for example, learning requires our synapses to change. And if our synapses change, the circuitry can change. Or the roads that I was talking about earlier. This ability of our brains to change in response to specific stimuli is called experience-dependent plasticity. We know that when we are young, plasticity is high, and as we grow older, plasticity decreases and synapses become more stable. Remember how effortless it was for you to learn a new thing when you were younger? And how much harder it feels now if you try to learn a new language or how to play a new instrument? Well, it is harder. This is because plasticity is age dependent. And this is not our brain playing tricks on us. Plasticity requires energy, and it just would not be optimal for us to retain that level of plasticity for our whole lives. Actually, we know that some disorders have alterations in plasticity. Okay, so we know our brain's connections can change. But what determines if they can change or not? We think astrocytes are playing a big role in how permissive the environment is for change in our brains. Scientists took young astrocytes, transplanted them into the adult brain, and they saw that this adult brain was able to change in the same degree that you would expect a young brain to change. We also know that astrocytes change with age, specifically in the genes that they express. Genes are like the instructions to form a tool, where the proteins are like tools. And a lot of tools are required for our brain to function properly and to change. So how can we study if astrocytes are involved in regulating experience-dependent plasticity? One way to do so is by focusing on the visual system. The eyes are the windows to our brain. This is more than a boring cliche. The eyes are literally connected to the brain. If we change what the eyes are exposed to, and we manipulate astrocytes in the adult brain, can we then study the changes in the circuitry in the part of the brain that is involved in visual processing? With my research, I found that after visual stimulation, astrocytes upregulate a protein in the part of the brain that processes visual information. This protein, or tool, is like an on and off switch for the expression of other genes. What's even more exciting is that I found these genes that are potentially being turned on and off in the astrocytes may be involved in synapse regulation. What I want to investigate is whether this protein is providing the astrocytes with instructions to stabilize synapses in adulthood. Specifically, I want to remove this protein from astrocytes and then see if I can increase plasticity in the adult brain to the level or the extent that you would expect a young brain to change. In summary, I study experience-dependent plasticity and how this may be regulated by astrocytes. With my research, I am able to manipulate the external environment and then look into the brain at the cellular and molecular level to find a link between experience and physical changes, and assess how these experiences may change the cells and how they interact with one another. This research is important and exciting because it challenges the well-established notion that plasticity is neuron intrinsic. And we know that in neurodevelopmental and neuropsychiatric disorders such as autism and schizophrenia, the way the brain responds and interprets the outside world is different. So having a better understanding of how different cell types help us transform sensory experience into information in our brain could help us develop therapies to treat these and other disorders.